preparing to stream. All right, and we are officially live. I'm going to share this on Twitter just because Zoom isn't always the most accessible to everybody if they have bandwidth issues and whatnot. So we always try to make it available via YouTube as well. And all right, we're right at 6 o'clock. So let's go ahead and get started. Hey, everybody, my name is Caroline Nickerson. I am with the SciStarter team, and I'm so delighted to be here tonight with my co-host, Deja Perkins, and our guest, Peregrine. We want to thank our supporters and our partners tonight. So this event is put on by the NC State University Citizen Science Campus, as well as SciStarter. And even though we are North Carolina focused, we are open to everybody. So all are welcome at these events. I like to kick things off each week by showing you your one-stop shop for citizen science. I guess I shouldn't call it a shop because everything's free, but this is scistarter.org/NCSU home. Here you can find information about the Citizen Science Club at NC State libraries, as well as a link to the main Citizen Science Campus website. This is also where we keep our season lineup and future episodes of Make It Count Mondays. We're here every single week, 6 o'clock Eastern on Zoom and on YouTube to make citizen science accessible to all and just have fun conversations and have a good time, frankly. This is also where you can discover a citizen science tutorial. You can click through it, do some interactive quizzes, you can watch some videos and just learn a little bit more about the who, the what, the how, and the why of citizen science, and you get a certificate at the end. And we also have a number of featured projects on this page. Coming soon is Wild Sourdough, but you can get started with Sourdough for Science. And there are a bunch of other projects too that you can do from home or you can do in your community, like Stall Catchers, COVID Near You, and Marine Debris Tracker. There's really something for everyone with citizen science. And we also have different lists. So like projects to do at home, projects to do on my computer. These lists were created by people from the NC State community. So students, instructors, and more of projects that they really enjoy of all the thousands of projects on SciStarter. And I know we have kind of diverse groups who come to these events. Some people have never heard of citizen science before. Some people are super pros. So we always like to give a brief definition of citizen science. One way we think of it at SciStarter is citizen science allows anyone and everyone to turn their curiosity into impact. So what does that mean? It means that if you're curious about pollinators, or you're curious about the night sky, or you really enjoy like thinking about the brain or other health issues, you're able to move science forward by volunteering. You can collect data, you can analyze data, you can be a part of the process of creating new knowledge without any formal training, just by following the instructions and joining projects. SciStarter is your tool to discover those projects. And as we were talking about before this show with the panel, North Carolina and NC State are a huge hub for citizen science. So across the entire state with institutions like the Museum of Life and Science and so much more, there's really amazing public participation and research going on. So we're grateful to all of you for being a part of it. So at this point in the show, I always pass the mic to my co-host, Deja Perkins, who kind of contextualizes things and gives an introduction to the topic of the day. Take it away, Deja.
Hello, everybody. My name is Deja Perkins. Welcome back to Make It Count Monday. This week, it's the week of Thanksgiving, which is one of my favorite holidays, mostly because my birthday is right around the same time. But I think Thanksgiving is a time where good food brings family together. And what goes better with mac and cheese, greens, sweet potatoes, and turkey than bread? That's right. Hold on to your stomachs because this week we are talking about bread. More specifically, we're talking about sourdough. So, before we get too deep, I want to ask, what is the food that you think about that instantly causes you to salivate? And while we wait for everyone to drop their answers in the chat, I am going to ask our on-screen audience what food it is that makes them salivate.
This is embarrassing, and I don't know if they're necessarily a food, but fruit gushers.
I love fruit gushers.
Yeah, I was going to say anything with citric acid in it, with that delicious flavoring. So I think definitely candies. I'm a big lover of mint, so like a candy cane is always what I go to immediately.
Okay, two candy answers.
What
You
about you,
know,
Deja?
I'm really not sure. Um, I think maybe sometimes like sweets like cinnamon rolls will cause me to, instantly just kind of drool a little bit, get a little pull. um, and yeah, so I mean that maybe sometimes even I mean for me, I think it's more so um, the sight in connection with the food. So like sometimes when I see like seafood or just whenever I'm really hungry, honestly. Um, so go ahead.
Oh, I was just gonna say, so and it's an adaptation, right? The amylase in our saliva that helps us digest things like starches. So it's like our body's evolutionary response to there being food. So that's really cool, and especially when we get talking about sourdough and the sourness, which makes us salivate. Just wanted to add that little piece.
Yeah, exactly. So that kind of brings me right up to the next point. Um, If you don't remember who the scientist Pavlov was, he was a psychologist who trained or conditioned his dogs to salivate to the sound of a bell. And so we often, as Peregrine mentioned, like we can get this response when we think about bread. And I am not going to lie to you all. I've always thought that restaurants serve bread to give us something to hold us over, right before our meals. But now I'm starting to think that it's a tactic that they use in order to prep us for our meals and get us to salivate a little bit more and get us, wanting more food from them. So, like Peregrine said, that saliva is our body's first wave of digestion. The amylase in our spit causes starch to break down into sugars. And so that's that I'm hungry, that evolutionary trait in response to food. And so, have y'all ever thought about why some foods don't elicit this response? So, if we think about saltine crackers and many store bought breads, they lack those acids that are produced in sourdough. And so, as a result, they tend to dry our mouths out instead of, getting us to salivate. So, if you think about when you're sick, that's probably why we do saltine crackers to kind of tamper down that notion to, upchuck everything. Um, but um, the moment that we take a bite of sourdough, the flavor receptors light up in our body and it starts to prep us for more. And so nowadays we can easily go pick up, whatever ingredients that we need in order to make homemade bread. Well, really about homemade anything. But our ancestors didn't necessarily have those advantages that we have today. They couldn't store yeast, and so they needed the wild yeast in the environment to ferment their dough. And so, what is it about the environment that causes us to be able to use these advantages to make food? I mean, really, it's something called a microbe. And a microbe is a microscopic organism. So it's an organism so tiny, it's unable to be detected by the human eye. So think back to maybe those classes in either high school or college where, you got to look through a microscope and look at everything under there to see like the structure of things. And so these microorganisms have been used since ancient times to make bread, cheese, yogurt, and my favorite, wine. um, and so for this project that we're talking about this week, we are super interested in the microbes that aid in fermentation. And so food manufacturers continue to use these microorganisms today to make a wide range of food products through fermentation. And so fermentation not only gives food a good taste, texture, but apparently also smell. And so it's causes changes that reduce the growth of unwanted food microbes and improves the food storage life and safety. So like I said, their fermentation is used to make a wide range of food and drink today. So if we think about it, yeast are used to ferment the fruits to make the wine, to ferment cereals to make beer, and bread, and flavoring in the form of yeast extract. So also we have this like filamentous fungi that are used in traditional processes for ripening cheeses and in the production of enzymes used in the food industry. And I'm not sure if this counts, but I have just started using nutritional yeast when I make my kale chips. And so apparently that's what's supposed to make them taste better. So I guess if we really think about it, this week we are getting into food science, which is great. Food science is the study of the physical, biological, and chemical makeup of food, and it looks into the causes of food deterioration and the concepts underlying food processing. So I say, let's see if we can try our hands at becoming food scientists this week. And I want to know how many of you all have tried the quarantine trend of baking, either during the beginning or, finally during the end. Caroline raised her hand. She said, yes, me. And so did Peregrine. I haven't gotten into that trend because I am a horrible baker. It's the one thing that I cannot do because I cannot follow instructions. So anything that involves a recipe is just really bad for me. I tend to just kind of cook by just, throwing things in the pan and shaking until I hear my ancestors scream, stop. So that's how I tend to cook. So if you are one of those people who have tried their hands at baking during quarantine, like you are on your route to becoming a food scientist, this project is definitely for you. And if, like me, you haven't had the time or just haven't been able to try this project yet, this is still the project for you, because right now, this week is the perfect time to try and make bread. I mean, get your family together and let's make bread, let's break bread together. So let's find out more about the science of sourdough. I am going to introduce our special guest this week. This week we have Peregrine Ratchi. Is that how you pronounce your last name?
Ratchi, you got it.
Awesome. Peregrine is an educator with the Museum of Life and Science. Peregrine earned a degree in anthropology from UNC Charlotte and has since found joy as an informal educator in the museum world, where they can teach authentically, inclusively, and experientially. A self-described educational generalist, Peregrine is especially interested in evolutionary biology and human origins. In their spare time, Peregrine likes to be outdoors and do experimental archaeology with lithics and other early human technology, which sounds really, really cool. Their teaching philosophy is, we've got some early human technologies being pulled into the camp.
I always try to have them on hand, so they're here, just in case.
Very nice. Peregrine's teaching philosophy is centered in cultivating skepticism and wonder as tools for seeking and understanding and making the world safe for human differences. So Peregrine is all about creating new scientists, which makes them the perfect guest for this week. Welcome to the show, Peregrine.
Thank you so much for having me, Deja. I'm really excited to be here.
Yes, we are super excited to have you here. So I guess as we kind of enter into the Q&A portion of the show, I want to know, when did you first get introduced to citizen science, or what is it that got you hooked on citizen science?
I kind of first came across the idea as an educator at the Museum of Life and Science, particularly in the lab, which is sort of a science outreach space, or at least in-person facilitation times. It's a place where we have educators talking to visitors who do different experiments and STEM activities and stuff like that. I was facilitating some of these activities, and I believe it was Wolf Watch was the first one that we did. And I was doing it with different visitors, and I was just thinking, finally, because it was finally just, I don't know, I guess as a kid in science class, I always felt like I want to do science, I want to do real science. And I guess I hadn't yet thought about how all science is real science, but that participatory science is something that everyone can do. It's real science in the sense that it's being recorded, written down, and being used to analyze information and create new knowledge. And it kind of enhances things that people do anyway, which is to use the scientific process to learn more about the world.
That's awesome. So for our featured and I love that you gave us that overview of participatory science. Could you kind of walk us through the featured project for today and a little bit of the context of, I mean, there's so much to cover, so I'll just let you take us on the journey.
For sure. So I'm not a sourdough expert by any means. Deja, I really related to when you were talking about how you're not super into baking, more into cooking, which is kind of more like maybe instinctive, or you just kind of use your intuition to do it. Following those instructions is always something that's been a little bit more challenging for me. But these instructions are kind of, there are very few of them. So it's kind of easy to follow along. But this sourdough project is something that originally started in the NC State Applied Ecology Department with an interest in the microbial world, because it's all around us. The microbial world is around us, it's on our bodies, it's inside of our bodies that help make our bodies work. And there's an interest specifically in yeast and what different yeasts do for us. So this little fungus, the yeast, that lives both on our skin and inside of our bodies and helps us make bread. And the question was, how do we make it? How do we make different kinds of sourdough starters? What contributes to a successful starter, or what makes a starter fail? And trying to get an idea of all the different diversity in the world of yeast, because there are so many, not just the kind that come in a packet in the store. There's yeasts all around us, and we're still just kind of scratching the surface of how many there are. So the original project was the Global Sourdough Project, and that was where they kind of solicited from all over the world, your sourdough starter, whether it be kind of an ancient one handed down from members of your family, which is something that happens pretty frequently, or one you just started. And then after that came Sourdough for Science, which is what you guys currently have on your portal. And it's a great project. It's really great for classrooms, especially. It really hits a lot of those science standards. And it's making a sourdough starter, making leaven in the classroom. And then this most recent iteration came right with the pandemic and lockdown. It was something that was kind of coming out anyway. It was kind of scaling the classroom version down for people to do at home. But then the perfect, in some ways, occasion arose for people to do this in their houses in a way that's kind of scaled down and only using things that you're able to find around your house or easily find at the grocery store. So Wild Sourdough, which is the most recent iteration of Sourdough for Science, thank you so much for linking that, Caroline, uses, it's kind of like a scaled down version of the larger experiment, where you're only using a little bit, we're talking like tablespoons and teaspoons of water and yeast. So what you need is, you need a clear container with straight sides, because a lot of what you're going to be looking at is the rise of yeast. So you have your container, it's clear. You have a little bit of flour, and I have just regular all-purpose flour. You just want to make sure it's not self-rising flour, because that already has the leavening agents in it. We're looking for, I think, a slightly slower process of rise. So we're waiting for the yeast in our environments to make this rise, not leavening agents. You don't have to use flour. You can use gluten-free flour, you can use barley flour, rye flour, all different kinds. In fact, they're really interested in the different kinds of flour and how that would affect the leaven. But you're going to need a little bit of this all-purpose flour. You're also going to need some water. And the water should be dechlorinated, which you can get dechlorinated water just by getting it from your tap and then leaving it out for like 8 hours. I don't know about you guys, but it's like signs in my house, like I have just like cups of water that I leave abandoned places. So I've always got some dechlorinated water on hand. You're also going to need some measuring spoons to help you measure. I just have a tablespoon and a teaspoon. And then for the initial process, I'm going to need a little spatula. I'm also going to need a rubber band and a little piece of paper towel. So to get started making your sourdough starter, making your leaven, you have to create sort of like a little home for the yeast to come and live in. So we have our kind of container, and we want to make a little house for the sourdough starter. So what we're going to do is we're going to take 2 teaspoons of our flour, I'm just going to take 1 teaspoon and 2 teaspoons. And then I'm going to put in equal parts water. I'm just going to put in 2 tablespoons. Did I say teaspoons before? This is tablespoons. 2 tablespoons of water. And then I'm just going to stir it up a little bit. I have a little stir stick here. So I'm going to stir up my mixture. And this is the beginning of it, right? This is kind of all we need to get started, attracting our own yeast to make our own sourdough. So super simple, a small amount of supplies to get started. And I think it's a really cool project because you're making a home for something that's alive. And it doesn't seem alive because it's just flour, it's just water, it's just like, it looks like toothpaste in there, right? But we're going to be creating a house for something that is living and in fact is living all around us. The next thing we want to do is use our spatula to scrape down the sides, because we're going to want to keep track of the rise. The rise is something where the yeast colony, as it grows, it will consume the sugar, or I guess it will break down the starches into sugar and consume those sugars. And as a natural byproduct, it will produce bubbles of carbon dioxide. Those bubbles are what makes bread fluffy. So unless you're making a flat bread, you need those bubbles of carbon dioxide. And as these yeast kind of migrate in from our environment, and there may even be some yeast currently inside of this container, because it's not a sterilized container or anything like that, it will grow. And they will start to consume that sugar and they will start to bubble and it will start to rise. And that's something where you can keep track of it by just looking, or you can use just a marker to mark. So I'm going to say this is day 1 right here. I'm going to put a little 1 next to it. I can either use a ruler to measure the height or I can just write on it. And then what I'm going to do is just cover it. I'm taking a little paper towel and I'm taking my rubber band. And I'm just creating a little cover for it. This allows oxygen to get in, it allows microbes to get in, but it keeps stuff like insects and other debris out. So this is the beginning of my sourdough starter. All I need to do is put it somewhere kind of warm and pretty dark. And the yeast will start to wake up. This yeast will start to get to work, eating and producing carbon dioxide and reproducing. And what I'll have to do now in order to participate in the Wild Sourdough project is for the next 10 days, I'm going to take this out at the same time each day. So it's 6:22 right now. If I put this away, tomorrow it's 6:22. What I'm going to need to do is feed it. It's like a pet. It's something alive. I need to be able to feed it. So what I'm going to do to feed it is to take off my lid, and I'm going to take out 1 tablespoon. So this is called, get ready for your new favorite name of something, backslop. Gross, but you can cook now. When this is matured and when this is bubbling and nice and leavened, you can use your backslop to cook. But right now this is just kind of paste. So I'm going to discard it. And it's something that can be composted or can be thrown away. But I've taken away 1 tablespoon because if I were to just add to it, first of all, the rise wouldn't be measuring the carbon dioxide. It would just be the amount of flour I was putting in. It also would just mean that we would have to feed it more and more as it got bigger and bigger. So I've removed 1 tablespoon. Now I'm going to feed it with the same stuff I made it out of. So I'm taking a little bit of my flour. I'm going to use 4 teaspoons. So that's like a tablespoon and 1/3. So I'm just going to take 4 teaspoons. So it's like I'm replacing what I took out, plus just a little bit more to grow on. Then I'm going to take my my water, my dechlorinated water, and I'm just adding 1 more tablespoon of that. Then I'm stirring it up, creating that nice paste again. And as soon as it's all combined, I'll scrape down the sides and I will mark the level. And that's essentially it. For 10 days, we're just feeding it. We're just looking at it. Oh, we are doing one more very important thing, which is smelling it. The way that yeast smells and as it grows, as it changes, is something that can tell us a lot about the different species and its different stages of progress. So different kinds of yeasts smell different ways. I know my first sourdough starter I described as smelling like a Parmesan armpit kind of bouquet. So not like great, but within a few days it started smelling kind of like pizza dough. So it changes over time as that microbial community changes and in many cases matures. It's kind of hard to describe smells, right? If we're talking about describe the part of our brain that describes words and the part of our brain that takes in smells, they're pretty far apart, and it's kind of different. It's kind of difficult to describe how something smells. But part of the Wild Sourdough project is this aroma wheel, and you can find that on the Wild Sourdough page. So you can look at this predetermined wheel and find the word that best describes it so that it can be analyzed more kind of systematically. So I'm going to say here on my imaginary day 2, I've scraped down my sides as best as I can. I'm going to mark my new level. It's probably about the same as my first one. And I'll smell it and think about how it smells and use that aroma wheel to describe it. Now all I have to do is put it back. Putting my paper towel back on, I'm rubber banding it, and I'm putting it in the warm, dark location. That's essentially it. There's a few things that you can try if you want. You can make 2 of these and put one in a closet and one outside and see how they develop differently. You can also introduce other variables, like one can be barley flour and one can be wheat flour, and you can see how that changes. There are some different ways that you can modify the experiment, and that's also on the Wild Sourdough page. That's the essentials of the project.
I actually have the aroma wheel right here. I believe you said earlier Parmesan armpit. Can I find that on here?
Well, no. Okay, that was of my own description.
Got it.
See.
We should lobby them to get Parmesan armpit added to the.
Well, there is fart. I don't think it was quite fart.
There's fart, and there's also sweaty feet.
Maybe it was more foot-y.
That's right. Feet and armpits. I think there's some overlap there for sure. I love all these smells. We got lemon. This is some good stuff.
Well, and it will kind of develop kind of a sour smell, right? That community of microbes, and indeed why we call it sourdough, is because the yeast kind of has this relationship with these other bacteria called lactobacteria, and they produce acid, and they kind of live together in their little community here, and they keep other bacteria, like harmful anaerobic bacteria that can make us sick, they keep them from being able to kind of make their home inside of this delicious starch and sugar concoction. So that's where we get it sour. Yeah.
That's really amazing to hear. So what do you think is next for this project? Like what will we learn from it? What will the research outcomes be?
That's a great question. When I asked Dr. Erin McKenney, who is one of the scientists who is analyzing this data, she's just really, really interested in fermentology. She studies not only sourdough stuff, she also studies different things. Well, she kind of pickles a lot of different things. I know that something that she showed us was she was pickling some tomatoes, and sauerkraut is another one that she's interested in. So I think she's really interested in this relationship between people and our food and how that's kind of grown and changed over the ages. And just kind of, especially what makes something succeed or fail is what she was saying is a really key factor. And part of this is not only compiling the data, but it's participating in science, right? So I think that this is something that is what participatory science is all about, right? It is about building new knowledge. It is about data, but it's also just about doing something and being part of a community of practice. Because I think that's what science is. You know, the data is something you dig around for and you use to create new knowledge. But science is something that you do. It's a tool that you use, and it's a group of people that you do it with. And it's even more amazing when it's not only people around you and people, in many cases, who you've never met, but people in different times than you. When we're thinking about sourdough starters that belonged to people of different generations, when we're talking about thinking back on the human legacy of using yeast, it's being a part of something bigger, of our own community, as we're learning about a microbial community.
That's really beautiful. And before I let Deja get a question in, I love that you have this parallel through history running through with the sourdough, one of our most essential foods. I keep on thinking about the article that you wrote, and I put that in the chat. And I'll add it to the NC State page as well. But I was wondering, is that kind of the thread you use to tell the story of this project? I'd love if you could speak to the storytelling aspect.
For sure. I think that, yeah, that article, which myself and my colleague Max Colley and also Dr. Erin McKenney, we wrote that together. And other than the appeal of it being bread, and bread is delicious, there's a reason that we essentially invented agriculture to make more bread. So the first breads were not made out of domesticated grains. We domesticated grains so that we can make more bread. So like, we love bread. It's just, it's written in our DNA. We just love it. So bread itself is very compelling. But definitely the kind of evolutionary like lineage and the kind of the mystique of sourdough over time is something that I'm really interested in. It being something that caused us to, it being a catalyst that helped us to invent agriculture, which totally revolutionized the way that humans live. I think that's fascinating. I think another one of the kind of story elements we used to talk about this one was the pandemic itself, because sourdough kind of became part of the zeitgeist. Everyone started making sourdough bread, and the big question was like, why sourdough? Why now? This is something that like people have always been doing. Why is it such a big deal? And if you think back, you know, we were running out of staples. You know, something like bread was something that was flying off of the shelves as people were going to like the one place you were allowed to go, to the grocery store at the very beginning of the pandemic and lockdown. And as they were out of bread, people were like, okay, well, if I can't buy bread, I can certainly make bread at home. And while flour was something, you know, you could get a little bit more easily in bulk, it was something that they could keep, stores could keep stocked. Yeast flew off the shelves, and self-rising flour flew off the shelves. So people were going, okay, well, if I can get my hold of the flour, can I still make the bread? I still need that yeast, but yeast is all around you. So you could continue to make your own bread. You could make just really a fundamental food stuff in this really strange, as everyone is saying, unprecedented times, without relying on the yeast from the grocery store and using the yeast around you. So it's something that you use your environment to create food for yourself, and in many cases, food for your family.
I love that. I tell people all the time that science is like one of the most important fields that, you know, we have today that people can get a career in. I tell people all the time that agriculture is life, and the fact that I can be at home and make something so essential and do science at the same time is just amazing to me. So I actually had a question that is related to something we kind of already talked about already, and that being the wheel of the different scents. So is it possible to still do this project and contribute if you can't smell? Because I don't have a sense of smell. And so I was just curious as to what the different scents tell you and how, I guess, your participation in the project might be altered from not having that sense.
Sure, and I appreciate you asking it, because I think one of the big things about this project is accessibility. So if you're unable to analyze the aroma itself, then tracking the rise is definitely something that you can do. There's a part of a previous iteration, more for classrooms, that also analyzes pH, so how acidic it is. So if you have access to something like pH strips, that's something that you can test the pH of your sourdough starter every day. But even if you don't have access to those, just tracking the rise, right? If I leave this in a cupboard, in a dark cupboard, and then I come back to it on day 2 or day 3, I'll start to see it bubble up. And tracking that change is something that I can use to contribute to the project. The aroma itself is I think kind of a fun piece, and a piece to kind of help you get emotionally attached to your starter. Smell is something that can evoke a really strong emotional response, such as disgust or delight, but I think a lot of times disgust, especially when we're talking about in the realm of like feet. But what that smell is telling us, if we can pick up on that smell, what it's telling us is just, it can tell us about the breakdown of the different kinds of colonies that are in here and the different species and strains of yeast that inhabit it. So we're learning more about different yeasts in different places based on the traces they leave behind, such as rise and rate of rise, and in many cases, such as the smell that they make on different days.
Yeah, that is really cool. I don't think I've ever thought about yeast having a smell, or that different smells can actually tell you things. And I thought it was interesting what you said earlier about the two sections of the brain being a little bit further apart, with association and smell, just because I ask people all the time to describe to me, like, oh, what does so and so smell like, and they have a really hard time trying to figure it out. So I thought that was kind of an interesting parallel. But I also wanted to know, I guess, maybe what's your favorite group to engage with when it comes to this project?
I think that a lot of times who I kind of aim at are the younger crowd. And in fact, since we can't do face-to-face facilitation in the lab right now, because it's not safe to do so with the pandemic, we've kind of shifted online, and we have a program called Lab at Home and Little Lab at Home for the kind of younger ones. And we first debuted this project on Lab at Home. And it's something that attracts kids. And I think a lot of times when parents see the newsletter and they see it on there, they're like, oh, that's interesting, I'll take that and have my kid watch it. But they're usually watching in the background. And I'm always aiming my language at the kids and engaging kids. But I think it's their parents who are really interested too. Their parents and their families want a way to engage with them and to connect with them. And that's something that we've seen with the Sourdough Project, is connecting intergenerationally. That's something that Dr. Erin McKenney commented on really quickly in the Global Sourdough Project. We had a sourdough historian who compiled a word cloud of kind of some of the different words that people used to talk about why they did what they did, and kind of who they were and why they liked doing this. And the biggest word in the cloud is love. And the next biggest word in the cloud is years. And the next biggest word in the cloud is try. So it's just sort of this beautiful picture of people doing things together over time, motivated with love, and also intrinsically motivated to try things again, even when they fail. That was the long way of saying kids, but also families.
I love that. Really quickly, on the topic of Lab at Home, could you tell us a little bit about, and this is, I guess, a little bit away from the sourdough, but all the museum education you've been doing, especially in the context of the pandemic?
Yeah, for sure. It's been really challenging, and especially in the very beginning, during the stay-at-home orders. If you're going to be doing projects with people, you can't tell them to go out and get a bunch of things. There was a day right before the stay-at-home order when I was able to go to the museum and grab a bunch of things that I thought maybe I could share with people. I had this whole big bag of stuff I just snagged from the lab, thinking maybe I can do something with it. But what we really wanted to do was have people do science with us, because I think, as I was talking about earlier, and as both of you have said, the whole point of like participatory science is participating, is doing it. I think doing science, practicing science and using it as a tool, doing it with people as a community, practicing that way of thinking is what it's all about. So we had to figure out a way to do these with people over the internet. So it's sort of a format a lot like this. It's a webinar. People talk to us through the chat, and I give them ahead of time, like a week ahead of time, I give them a materials list so they can grab some of the stuff that they need, and they can do it along with us. And a lot of people do. I was surprised. I was sort of waiting for, I don't know if you all watch any like YouTube and you watch the like DIY stuff, and I'm like, I'm never gonna make this, but I like watching you do it. But a lot of people do it along with me, and kind of tell me about what's happening with theirs. And it's a really cool way to connect. I think especially in the lockdown days, those were a really, really amazing way to connect. And we saw a lot of people who really wanted to connect in that way.
Related to that, kind of the human element, could you tell us a little bit about your community? Do you have people from all over the world who are participating in the project? Like, how is it kind of resonating in North Carolina? We'd just love to hear about the community and citizen scientists working with you.
Yeah, for sure. So it's largely local. It's largely people who have been to the museum, but we've had people who live locally, and they still tune in, like when they go on vacation. We had someone who was like, "Oh, we're at this was over the summer." Someone was like, "Oh, I'm at the beach today, but I'm still hanging out." And we really have goals of involving families who live in different places all over the country, because the one kind of silver lining about things is that you can in many ways connect with people more easily digitally in some ways, right? Because we still have to contend with things like the digital divide, and that's something that I'm keeping in mind as I'm creating all this digital programming. But at least I can try to engage families that live in different places, and just kind of like people outside of our community as well.
Yeah, I think I really love how easy this project is. I mean, just watching you do the demo, it really broke down, I guess, some of that hesitation that I had about jumping in and trying the project. So just, a little sidebar, I can't wait to go home and do this project with my many little siblings that I have. They love like doing little things like this. And I think that this is a great week. We all kind of coming together to make either like Thanksgiving food or whatever. I think this is a great time to try and bring people together through science. So is there anything you would tell people who may, I guess, be nervous about getting started in this project, but still kind of want to participate?
Yeah, and Deja, I'm so excited to hear that you're interested in trying, because that's like the best thing to hear. That's the greatest thing to hear, because sometimes starting is the hardest part. So for anyone who's nervous to start, I want to tell you that even if you fail, and we like to use failure as kind of a dirty word, but it's a really important one when we come to science, because failure is still data. This is something that when working with the scientists who developed this project, they're saying, even if your sourdough fails, even if you think it has failed, it's still important information. Please still report it. Please still do it, and please still try. The great thing about kind of persisting through failure, right? That's something that gets you through in other parts of your life too. It's one of the other reasons I think science is so important, is because if we can learn to contend with failure and just accept it, that in a scientific experiment, that helps us in other parts of our lives too. So I would say for anyone who's nervous, first, you saw how relatively simple it was to get started. You also know that it's not uncommon to fail. A lot of people have this issue. I had this issue. I love talking about failures. I definitely, my first sourdough starter was going great the first couple days. Then I missed a day feeding it, and I thought, oh, it'll be fine. I'll just feed it twice next time. It didn't do so hot after that, so I failed. It didn't do what I wanted it to do. It wasn't smelling right. So I tried again. I figure it's only a couple tablespoons of flour. It's only a couple tablespoons of water. So that's what I would say to you is let failure be okay with you. Let it still be data, because it will be for the scientists who are collecting that data. And just keep trying. And if you have to motivate yourself with anything, motivate yourself with the idea of bread, because the bread will come.
Have you eaten the results of any of these experiments?
I have not made my own sourdough out of my own sourdough starter. So I've made bread, but I have, this is another shameful secret. I have been using some store-bought yeast that was, it's actually pre-pandemic yeast that was sitting in the lab, and I've actually just kind of used a pinch of that here and there. My own sourdough starter, currently, I restarted it recently, but I put it in the fridge and it's kind of in stasis right now. But I am really motivated, and part of it was coming and talking to you all. I am really motivated to get it started again. That's the thing too, is it's going to be there, and it's also a really forgiving thing. If you want to restart it and try again, it'll be there. The yeast is all around us. It's not going anywhere.
I guess I was wondering along those lines, what have you all done with the data so far that you've collected? And maybe, I guess, next steps with the project?
Yeah, so the Global Sourdough Project, the very first one, has concluded. That data has been compiled. Sourdough for Science is ongoing, and Wild Sourdough, which is the newest, the kind of pandemic-friendly version, is still ongoing. I think when I was talking to Erin, she said there's something like 1,700 entries so far, or individual like starters that have been reported so far, and at least 100 of them have been in the last like 2 months. So people are still doing it. People are still reporting their starters. So it's something that's ongoing, and it is something that they're still working on analyzing. I know that, again, talking to Erin, she was saying they're getting a whole lot of press about it because I think it's still part of the zeitgeist, part of the times. And while it's great to spread the word about it and get more participants, it's a little bit harder to analyze data when you're doing so many different presentations. So right now it's kind of, the focus is on encouraging more people to do it, making it more accessible for people to learn about it through various different means. I know some people involved with the project have been creating really cool things like yeast coloring pages and like a yeast name or a sourdough starter name generator, just different things to get people engaged. So it's kind of in the growing phase, I guess we could say. That data has yet to be all compiled.
I think you got me thinking there. I feel like it would be so cool for families out there. Like if you can't get together in person for the holidays, like if you just did a family-wide like sourdough project. And so each household, making their own little sourdough starters and seeing how it differs in everyone's households, no matter, like I'm here in Raleigh, but my family is in Chicago, or having, just family either in different states or family in different parts of the city, and how it, how their sourdough starters differ. So I think that would be so cool. Just anybody out there listening who wants to try that.
Please do it, because your sourdough is a part of you in some ways. I have yeast on my body. If I'm making my sourdough starter, it's not unlikely that part of the that maybe some of the strains of yeast that live in here are my yeast, right? Or they're yeast from my home. If you can't visit my home, you can have the yeast that lives in my home in my sourdough bread, right? So it really does that. If there's a piece of you. Similarly, if you're kneading your eventual dough, you're also putting additional strains of yeast on there just from your hands.
And in the chat, someone put the link for the sourdough name generator. So I say, let's do it right now. Ready, everybody? All right, we're about to name it. It's Snowloft Joel. All right, that's their name.
Snowloft Joel. I'm going to write that on the paper towel.
I'm going to reply to our Twitter thread with this live event with hashtag sourdough name generator. So stay tuned, everybody. And that being said, for anybody watching, or if you're watching on YouTube later, or you're listening to this on the podcast, let us know how you're doing this on Twitter. It's always really inspirational for me, especially in these times of social distancing and we're not physically together. I love engaging with you all online and getting inspired by the citizen science you're doing. So we'd love to know what you've named your sourdough starter and how you're doing wild sourdough. I guess to kind of close it out, because we have an audience of a lot of people who maybe want to do similar work or who are real doers, I'd love to hear your advice for people who want to run programs like this or want to get started in inspiring others to do citizen science.
For sure. There's a lot of really amazing kind of infrastructure out there. So I think the first thing I would do would be to say, check out the SciStarter page. A lot of local institutions like the Museum of Life and Science has their own portal. And I'm not sure if you already put that one in the chat, but the Museum of Life and Science has its own portal of stuff, of different citizen science projects that we're doing. There's a lot of infrastructure out there, so you're not like just having to do it all yourself. There's communities of people who are already doing it. There's lots of different resources, lots of different FAQs, lots of different things you can look at to help you get started. So once you're interested in getting started, there's a lot of stuff out there to help. And then I would say the next most important thing that you could do is find somebody to do it with. Because science is a community of practice. So finding someone to do it with and kind of getting other people involved is something that grows your own investment in the project. Because now you can't just, say, yeah, I gave up on that and nobody knows about it but me, so it's okay. When you have your friends and family asking after, oh, how's your sourdough starter doing? That maybe helps motivate you and keep doing it. But also you have someone to share your experiences with. And I think that's what it's all about too, right? Is as we're doing science, as we're collecting this data, as we're contributing to new information, we're also experiencing things for ourselves. We're getting to meet people. We're hearing from their own successes and failures. We're growing our relationships with our community. So, so check out all of the amazing resources out there. Find people to do it with. there's definitely, especially when it comes to sourdough, tons of people who are just nuts about sourdough, and they love to share information. That's something that we found through this project, is sourdough people are so generous. They really love, I think Dr. Erin McKenney mentioned something about during the pandemic, there being a community effort somewhere where they were stapling bags of sourdough starter to telephone poles to share it with people. So that's something, yeah, like just literally offering people the materials themselves to get started. Yeah.
Awesome. I love that. Go ahead.
I was going to say too, if there are any particular projects you are interested in, or if you're not sure where to get started, we're definitely doing some really cool ones at the museum that are centered around climate and kind of checking out some of the different things that are going on. We in particular have one about extreme precipitation events, and this is the perfect year for it, as the wettest year on record. I'm going to go ahead and you can check that out on our SciStarter portal, which I'm going to put up right here just in case. And then I also wanted to mention, we recently concluded our Climate Conscious NC project. So that's another one where you might be able to kind of, in some ways, join a community in hearing kind of people share their experiences with the project, and that can help inspire you to pursue the ongoing projects as well.
Yeah, really quickly on that, because we have a little bit of extra time. The Climate Conscious NC, and I urge everyone to go to the link in the chat that Peregrine put to explore that a little bit more. But I think that was really inspiring, in that you don't have to be, a climate expert to start these conversations. So for people who want to bring citizen science into their programming, or they just want to get their church or community group or whatever you're a part of started with these sorts of things, just start the conversations. Like, invite experts in to speak with your community, or just bring in some additional videos or additional content yourself that you're able to find through these trusted sources, and just start doing it. I think this was really inspiring for me tonight. Thank you so much for joining us and talking to us about not being afraid of failure and just putting yourself out there. And the main thing about citizen science is just learning by doing and starting. Deja, do you have any questions to finish us off? Oh, go ahead, Peregrine.
Oh, I was just going to say that, and just a little piece about how the great thing about a lot of times working with kids is they don't know that they're not supposed to do something. So something that I think holds us back is like, well, I'm not a scientist. I don't know if I can do science. I don't know enough. I haven't done years of research. And, while we always appreciate the insight of experts, science is something that you do. It's a tool that you use. It's something that I think made humans human in the first place, right? As we adapted to our different environments, as we experimented with different types of tools, including biotechnology like yeast. It's something that makes us human, and something that is essential for our continued survival. So do it because you can, because it's there, and because you're human.
Yeah, that's awesome. Thank you so much for joining us. I've learned so much today, and I'm really excited to get started with this project. I definitely think this is it's probably one of my favorite episodes that we've had so far, and I'm super excited to get started in learning more about sourdough.
Deja, during the holiday special, we should both show our starters that we've started and compare and contrast. That's my challenge to you.
I'm totally ready for that.
Great. And so, oh, sorry, go ahead.
Just gonna say, I'd be really interested in hearing about that as well. That sounds awesome.
Yes, join us for the last episode of the season. So to kind of close things off, we urge you all to go to scistarter.org/ncsu-home. I'll be adding all the links from the chat here tonight for you all to discover. And you can also watch past episodes of Make It Count Monday, see what's up next, including the Christmas holiday special coming from Deja and I. We'll compare our starters. You can find different projects to do, a tutorial, and so much more. Peregrine, do you have any words of wisdom or calls to action for us as we leave this evening?
I think I might be out of words of wisdom. You've
given us a lot tonight.
Just thank you all for being here, and thank you all for everything that you do to inspire people to be the scientists that we all are. This is Snelloff Joel, and myself signing off.
I love it. Well, have a great night. Thank you again. Bye, everybody.
Thank you both. Yeah, thanks so much, friends.